Hello everyone and welcome back to Biology with Mrs. Evans. We have finished our discussion on basically how proteins are made using messenger RNA, and we also talked about DNA replication. Up until this point, we've assumed that everything is basically copied flawless, and there are several basically safety checks that the cell goes through in order to make sure that DNA is being copied correctly, but mistakes do happen. And a mutation simply is any change in DNA, and there can be several causes for that. It can be exposure to ultraviolet light, radiation, all kinds of things. So anything that causes a mutation, we call a mutagen. And there are two big categories of mutations. One is a gene mutation, and that is a mutation that only affects a specific gene. And there are two types of gene mutations, which we're going to talk about in a second. And then chromosomal mutation is a mutation that affects the chromosome. So that can affect one or more genes depending upon where they're located on the chromosome. And then the significance of mutations, sometimes there's a change, but we call it a silent mutation because nothing really changes other than the base that got substituted. And I'll show how that works in a moment. And then sometimes the mutation is significant where it can be basically a catastrophic mutation. So sometimes the protein doesn't even get made at all. So we're going to look at that here in the next slide. So gene mutations, again, it is a mutation that only affects one specific gene. So there are two types. One is called a point mutation, and the other type is called a frameshift mutation. So point mutation, everything basically this box right here is talking about point mutation. So a point mutation is when a gene, basically, not a gene, but a base, nitrogenous base, is substituted for another. So you see, this is what the DNA sequence should have made. And then down here, it's showing you that if you look at the DNA sequence, this T got substituted for this C. So the result was this particular amino acid was coded for instead of this amino acid. So it's called a point mutation because the only thing that changed is right here, basically where that mutation occurred. Everything else is left the same. So that's why it's called a point mutation. And then a frame shift mutation, again, is another type of gene mutation, and that is caused when either a base is added incorrectly or a base has been basically deleted. So remember when I talked about how a ribosome reads the messenger RNA, it reads it in chunks of 3. So if you add or remove a base, it's causing that frame basically to be shifted. So if you look over here on the right, this is a sentence, the fat cat ate the rat. So if you add or remove a base, it's going to shift that chunk of 3, and this one here shows you what happens if you happen to delete this H. So instead of being a sentence that makes sense, you now are left with basically a jumbled mess. And if that happens, sometimes the protein won't be made at all, and sometimes it can be a catastrophic change. So, and we call that basically a missense or a nonsense mutation because nothing can come of it. And then point mutations, sometimes we refer to them as a silent mutation because yes, it may have changed a base, but the amino acid didn't actually get changed. And I'll show what that means here in a second. So remember the codon chart. Remember, this is a codon chart. It tells you what amino acids basically are coded for by the different codons. So remember I said a point mutation sometimes can be what we call a silent mutation. So if the change happens in, let's say, the third nitrogenous base. So let's say this is supposed to be, well, let me get my pencil. Let's say this was supposed to be UCG, but instead it becomes UCA. So let's see, what would that have coded for? So let's review. Remember, we go to our first nitrogenous base, which is here. Then we go over to the second one, which is here, come to where they meet, and we drop down to G, so that would code for serine. And then let's see what the mutation would call for. So again, here's our first base, here's our second base, here's where they meet, and sure enough, it's still going to code for the same amino acid. So that is what we would refer to as a silent mutation. Yes, there was a change, but it didn't change the outcome as far as what amino acid was coded for. So if it happens in the second and third base, that is typically sometimes what you'll see is there is actually no change. But if it changes this first base, it has then that's when you will see a different amino acid is most likely going to be coded for. So instead of saying UCG, let's say this time it codes for ACG. All right, so let's see what that codes for. So here's our first base, A. Here's our second base. Let's go to where they meet, which is this box. And we're going down to the G, which is right here. So this time it's going to code for threonine. So if it happens in the first base, you can see it can code for a different amino acid. But a lot of times if it just happens in that third base, it can still code for a lot of times the same or the correct amino acid, and that's why we call it a silent mutation. So chromosomal mutations, they are different than gene mutations because gene mutations is simply happens to basically the nitrogenous bases. A substitution results in a point mutation, and then a deletion or an insertion of a nitrogenous base is going to result in a frame shift mutation. And then chromosomal mutations have to do with the chromosomes themselves. So remember we talked about chromosomes and we talked about genes being located on chromosomes. So this is a chromosome that has basically 6 genes located on it. Gene A, gene B, gene C, so on and so forth. So a deletion is one of the genes has been basically deleted. So you can see that the gene B has been removed. So chromosomal mutation affects the whole chromosome itself. So this specific gene has been removed. A duplication just means that one of the genes has been duplicated, or it can be more than one. Sometimes it'll be 2 or 3 genes that have been duplicated. So if you look at, this is the normal chromosome, but over here this is what we have. So you can see that gene B has been duplicated. And then inversion is basically part of the chromosome has broken out and basically flipped. So instead of being the correct order A, B, C, D, E, F, we now have this order here. So you can see that the B, C, D, E has basically flipped itself. If you fix it, flip it back, B would be in the correct order, then C, D, E, and F. So an inversion, part breaks out, flips, and then basically gets reinserted back incorrectly. And then translocation basically is when pieces of a chromosome or 2 different chromosomes can break off and end up on another chromosome, and that happens during cell division. We're going to talk about that when we get into Unit 8. So you can see you have 2 separate chromosomes here. A, B, C, D, E, F is one, and then here's your second chromosome. But because they're so close together, when basically cells are dividing, pieces can actually break off and end up on another chromosome. So a translocation, when basically entire segments of genes can relocate. So you can see that GKL is now on this particular chromosome, and DEF is now on this chromosome. So chromosomal mutations affect the chromosomes themselves, and gene mutations only affect a specific gene. So mutation review, remember there are 2 types, 2 broad categories of mutations. You have a gene mutation, it only involves usually 1 or 2 nucleotides, and it only affects that specific gene. And an example of a gene mutation would be people that have sickle cell anemia. So you're going to see this in a lab that we're going to do. Hemoglobin is, remember, the protein that is in red blood cells, and a change in the amino acid sequence causes their red blood cells basically be deformed. And you'll see a picture of that when we get into, I think it's Unit 9. So an example of a gene mutation would be individuals that have sickle cell anemia, and then chromosomal mutations are mutations that affect basically whole chromosomes. So again, some mutations can be silent, meaning that there was a change, but it didn't change the outcome. The amino acid is still coded for, basically, that was needed to be coded for. So we call those basically a silent mutation. And then remember, we call a frame shift mutation, a lot of times we'll refer to that as a missense, because a base has been added or deleted and it causes the shift in the reading frame. And a lot of times, sometimes that shift, the ribosome never reads a start codon, so the protein is never made in some of these. So know the different types of mutations. Know basically the outcome, what happens in each. And then the last thing that I want to talk about is mutations are not always a bad thing. Sometimes they're a good thing. An example of that is bacteria. Bacteria replicate so frequently and with such speed that mutations happen pretty frequently for them. And you hear a lot in the news, we have bacteria that has become resistant to the antibiotics that we are using. It's because their genetic code mutated and allowed them to be resistant. Some plants have a mutation that allows them to resist bugs basically from eating them. So some mutations are devastating and other mutations are beneficial for the organism. So that concludes mutations. I hope you have a great day and I'll see you in class. Bye.